Hello and welcome. This is Columbia Basin Research's third video in a multi-part series on using Program Roster. Program Roster is a program created by Columbia Basin Research at the University of Washington's School of Aquatic and Fisheries Sciences. Program Roster models the life cycle of specific salmonids from the initiation of the juvenile migration as smolts to the end of the spawning migration as adults. In this third video, I'm going to explain some of the theory behind Roster. Future videos in this series will work through selecting the proper model and working through some of the issues you might run into with settings, file formats, and data sources, and offer some examples on common issues and how to avoid them. Alright, so in our previous video, we worked through defining your model, setting up your parameters, and running your estimation routines. Today, we'll talk about the theory behind Roster. Historically, juvenile and adult pit tag data have been analyzed separately and transported smolts have been removed, censored from analyses to estimate smolt survival. The results of these separate analyses can be inefficient and deceptive. A pit tag release recapture approach was developed that models the life cycle of Pacific salmonids from the initiation of the juvenile migration as smolts to the end of the spawning migration as adults. This model jointly analyzes juvenile and adult pit tag data, including transported individuals. Multiple performance measures for monitoring the juvenile and adult migrations and assessing recovery efforts are produced, including estimates of juvenile in-river survival, ocean survival, adult in-river survival, transportation effects, for example TI and D, SAR, and adult age composition. This model is appropriate for spring and summer Chinook salmon, sockeye salmon, and steelhead. Because the model does not accommodate the overwintering behavior of fall Chinook salmon, data from this species should not be analyzed with this model. The life cycle model primarily uses detections of pit tags at dams, but can also use detections at hatcheries or spawning sites if available. The model uses branching migration paths to capture the age structure of returning adults. Here in this figure, we can see a schematic of the migration paths used by the life cycle model. Survival S and detection probabilities P can be estimated at each detection site. Joint survival and detection lambda can be estimated at the last site. Smolts in the release group move downstream along a single path, but branch into one of several paths in the ocean based on maturation rates and transport history. Each branch represents a particular adult age class. These are kept separate throughout the adult spawning migration. Roster can accommodate up to 5 age classes. In addition to multiple age classes, the life cycle model incorporates transportation and can estimate transportation effects directly. Adult detections of transported fish are used to compare the return probabilities of transported and non-transported fish. Here in this figure, we can see a schematic of the migration paths of the transported and in-river fish. Each ocean and adult path represents multiple years. The model also accounts for censoring of juveniles and adults due to known removals at dams. This table here lists the parameters used by the release recapture model and estimated by Roster. Parameters are estimated by maximum likelihood, with the desirable properties of asymptotic, unbiasedness, efficiency, and normality. All combinations of the basic model parameters are also maximum likelihood estimators. It is possible that the effects of juvenile transportation extend through part or all of the adult migration. For example, juvenile transportation may affect adult straying rates. This would result in different adult in-river survival probabilities for transported and non-transported groups. Roster can fit a model that estimates the effects of juvenile transportation on upriver adult survival, detection, and censoring probabilities. If transportation effects really end at the first adult site, however, the extra complexity in the model that estimates upriver effects will detract from the information that can be learned from the dataset. Thus, the user can tell Roster which adult site should represent the extent of the effects of juvenile transportation, and by using model selection tools, for example the likelihood ratio test or AIC, determine the model with the appropriate extent of transportation effects. The user tells Roster what the extent of the transportation effects should be by changing the value in the extent of transportation effects window. The default value for the extent of transportation effects is the first adult site, because this relatively simple model is generally easier to fit than models that incorporate upriver transportation effects. Because juvenile and adult sites are numbered sequentially, the first adult site is one or more than the number of juvenile sites. With 7 juvenile sites, the extent of transportation effects value will be 8 by default. If a site is included, whether juvenile or adult, it must have detection capabilities. For example, for a data set with adult detections at Bonneville, McNary, Ice Harbor, and Lower Granite, Roster by default assumes the transportation effects end at Bonneville and that the adult survival, detection, and censoring probabilities upstream of Bonneville do not depend on the juvenile migration method, transport, or in-river. If the user wishes to investigate transportation effects on the adult migration up to McNary, then the value in the extent of transportation effects window should be changed to the index of the second adult site, for example to 9 if there are 7 juvenile sites. If the user wishes to investigate transportation effects through the entire adult migration, then the extent of transportation effects value should be changed to the last adult site, Lower Granite, site 10 if there are 6 juvenile sites, and 4 adult sites. After changing the value in the extent of transportation effects window, the default model must be loaded in order for roster to use the correct model. The model parameters for adult survival, detection, and censoring vary between transport and non-transport groups up to but not including the site that is identified as the extent of transportation effects. Thus, if transportation effects are measured to the first adult site, Bonneville, then only ocean survival varies between transport and non-transport groups. If transportation effects are measured for the second adult site, McNary, then detection and censoring probabilities at Bonneville and survival between Bonneville and McNary vary with the transport group. Upstream of the adult site identified as the extent of transportation effect, in-river survival and detection are assumed to be the same regardless of juvenile migration method, transportation, or in-river. By default, roster does not differentiate among transport groups in estimating upriver adult parameters. However, the expert user may modify the model definition file in order to compare, for example, upriver adult survival parameters for the different transport groups. Estimates of the model parameters and the performance measures are directly applicable only to the tagged fish in the release group. However, it is typical to assume that the tagged release group is representative of the untagged fish from the same migrating populations. If this is true, then the estimated values of the model parameters and some of the performance measures may be applied to those untagged fish. Note that the validity of such non-statistical inference depends on the degree to which the tag group represents the untagged population of interest and is independent of the statistical model behind roster. Other performance measures must be modified for untagged fish. In particular, any performance measure that depends on the conditional transport probabilities, for example TI, must be modified for untagged fish, because tagged and untagged fish experience the transportation system differently. In general, all untagged fish that are bypassed at a transport dam are transported at the dam. However, some tagged fish are diverted from the bypass system back to the river rather than to the transport barges for study purposes. This means that for tagged fish, the conditional transportation probability is usually less than 1. For example, TI is less than 1. But for untagged fish, the conditional transportation probability is usually equal to 1. Letting TU be the conditional transportation probability for untagged fish, we typically have TU equal to 1. It is possible that only a portion of the untagged fish in the bypass system are transported, in which case TUI will be less than 1. For example, transportation may be operated only on alternate days, which would be consistent with TU equals 0.5. Performance measures derived explicitly for untagged fish use the values TUI rather than the model parameters TI and are labeled with the superscript U to indicate untagged. Otherwise, these performance measures are identical to those derived for tagged fish. The TUI values cannot be estimated from the tagging data, but must come from other sources. The default values used in roster are TUI equals 1, SE equals 0 for sites where tagged fish were transported, and TUI equals 0, standard error equals 0 for sites where no tagged fish were transported. The user may change both the TUI and the standard error values on the roster interface. It should be noted that the direct statistical inference of performance measures using TUI applies to the fish in the tagged release group had they been treated as untagged fish. The default performance measures estimated by roster are defined both heuristically and analytically in terms of the model parameters. In order to address the needs of as many users as possible, it has often been necessary to create several versions of the same type of performance measure. For example, consider a measure of transportation effects. It is possible to measure the effect of transportation from a single transport dam, either in the context of the entire transport system or else isolated from that system. Alternatively, the effects of the entire transport system may be measured with all transport dams considered. The transport effect measure may be either a relative measure, ratio, or an absolute measure, different. The effect may be measured on return probabilities only to the adult site that have been identified in the model as the extent of the transportation effects E, or the way to the final adult site V plus U. The measure may be calculated for tagged fish or untagged fish, that is, tagged fish treated as untagged fish. And finally, the measure may or may not include jacks. This gives us a multitude of different transport effect measures to choose from. The program roster manual goes through most of the different types of measures explicitly. It can be found at cbr.washington.edu/analysis/apps/roster. The manual specifically looks at types types of measures including transportation effects, including both dam-specific, single dam, and system-wide, differential or delayed mortality, the ratio of post-Bonneville outmigration survival of transported fish to that of non-transported fish, juvenile survival, probability of surviving from the first detection site, usually Lower Granite Dam, to the last juvenile detection site, typically Bonneville, for non-transported fish, ocean return probabilities, probability of returning to the first adult detection site depending on various metrics, transported, non-transported, etc., adult return age distribution, probability to adults returning to a particular site in each of the adult age classes depending on various metrics, adult upriver survival, survival from Bonneville to Lower Granite for adult fish depending on various metrics. A more in-depth look at these metrics and their mathematical backing can be found in the manual. This has been Lesson 3 on using Program Roster, in which we explain some of the theory behind Program Roster. Future videos in this series will help you select the proper model, as well as offering various hints and tips on getting the most out of the program. We hope that this video helps you get the most out of Program Roster. Again, if you have any questions or comments in regard to Program Roster, you can find the user manual at cbr.washington.edu/analysis/apps/roster and fill out our user satisfaction survey there as previously detailed. You can also contact Columbia Basin Research at web@cbr.washington.edu. Best of luck with your analyses, and thank you.